Hi, I'm Matthew Wells, and this is my talk for the American Geophysical Union Fall meeting on vertical oscillations of the thermocline caused by internal waves modify fish distributions in lakes. So this is primarily work by Brian Flood, who recently graduated from his PhD thesis at the University of Toronto, and Jill Brooks, who will soon defend a PhD at Carleton University, Ottawa. There's a number of other government collaborators I've listed on the last slide. So when you look at fish, they have specific preferences for dissolved oxygen and temperature, and so you tend to find them at particular depths in thermally stratified lakes. In temperate lakes, we often classify fish into three categories. Those are the warm water fish, cool water fish, and cold water fish. And on this picture here, I've got some examples of fish we typically find in the Laurentian Great Lakes. So as the name suggests, during summer, you tend to find warm water fish in either warm littoral zones or the very surface of lakes. Cool water fish will be generally located above the thermocline, and then the cold water fish located in the cold waters beneath the thermoclines. So in my talk today, I'm going to look at two particular fish, a walleye, which is a warm water fish generally found above the thermocline in lakes, and cisco, which is a cold water fish found below the thermocline. So Hamilton Harbour has been extensively stocked with walleye to replace the fisheries that were depleted by the industry over the last 100 years. Generally find them above the thermocline in lakes. Cisco, on the other hand, they want very cold oxygenated waters, and you generally find them below the thermocline in Lake Simcoe. So a big question for both these fish is what happens when the thermocline's moving up and down? So to measure where fish are in lakes, the most robust way to determine the number of fish and where they are in the lake and the composition of the populations is to do a hydroacoustic survey. So we did this with my colleagues in Lake Simcoe using a transducer attached to the boat. The boat would sail at night time when the fish weren't schooling around Campbellfield Bay, and based upon the backscatter from the acoustic beams, we would determine using proprietary software which fish were in the lake, how many fish, and the age distribution of fish. So the raw data you get out is shown on the right, and once you run this through the software, you can work out how many tens of thousands of fish are in Lake Simcoe. This is a great technique. The only problem is it's very time consuming and expensive. You get good spatial information, but just at one point in time. So a new technique that's becoming quite popular in the Great Lakes is to do acoustic telemetry. Here you get long time series of what fish are present and what depths there are at particular points in space. So it's a complementary method to the hydroacoustic surveys. So each fish has a tag in it that's been surgically implanted. This tag sends out the fish ID at discrete intervals, and in many cases, the depth the fish is at and other variables as well. So in the Great Lakes, this is a collaborative effort with many institutions in Canada and the US, coordinated by GLATOS. There's over 1,000 receivers and 10,000 tags fish. This is giving another view in the dynamics of fish in these large lakes. So during summer, in a large lake that's stratified, you'll have a layer of warm water over cold water. However, it's not stable. Every time the wind blows, this warm water is pushed downwind, so you get downwelling at one end and cold upwelling at the other. When the wind stops, this relaxes and you get an oscillating movement of the thermocline going back and forth. What this implies is that at the edges of lakes, there's a region where there's great thermal variability, and in the center it's very stable. Now to study this, we looked at two lakes, one to the north of Toronto called Lake Simcoe. It's 50 kilometers wide, and we're particularly interested in the embayment on the west side, Lake Campbellfield Bay, which is about 17 kilometers long, 45 meters deep. This is where most of the cold water fish habitat is. The other region of study is Hamilton Harbour, which is an industrial harbour. It's had a legacy of pollution. There's no oxygen in the bottom of the harbour. We would like to know what fish can survive and thrive in the warm upper waters of the bay. So here they are side by side, Campbellfield Bay on the left, we're looking at the deep 45-meter region below, and Hamilton Harbour on the right. They both stratify in summer, they both have a strong stratification. Hamilton Harbour has almost no oxygen below the thermocline, and Campbellfield Bay has some oxygen below the thermocline. So in Kenmanville Bay, my colleagues did spatial surveys using hydroacoustics during the summer of 2015. And in Hamilton Harbour, there were dozens of fish with acoustic tags that were the spatial habitat usage was determined with fish telemetry in summer of 2016. So starting with Hamilton Harbour, if you go out in the centre of the harbour and measure the time series of temperature, you get this classic picture of a dimictic lake. On the top, I've got a picture of temperature. On the bottom, dissolved oxygen. Both have the y-axis for depth and the x-axis for time. So in a dimictic lake, you expect to see isothermal spring water column profile, stratified summer period, and an isothermal fall period. During the summer period, it's warm on the top, cold on the bottom, shuts down the supply of oxygen to the deep waters. And so because there's a large sediment oxygen demand, Hamilton Harbour has almost no oxygen below the thermocline, as shown on the bottom. And so this means any fish that can survive in Hamilton Harbour have to be able to cope in the upper water column. And so this is why there's a decision been made to stock Hamilton Harbour with walleye that prefer these warm waters. Now, what we'd like to know is what happens at the edges of Hamilton Harbour when you get these upwellings. So in a computer simulation Brian Flood did, he made a model of thermal stratification in Hamilton Harbour and looked at what happens when the wind blows from the west. Hopefully you can see in this image that when the wind blows, that stable stratification, the red water above the colder green and blue water is pushed downwind. And so regions at the edges of the harbour will experience quite big swings in thermal stratification. And if there's oxygen stratification, we'll see big swings in the dissolved oxygen levels. Here is two pictures of a time series of temperature profiles at the east end of the harbour and the west end of the harbour. And unlike the pictures in the middle of the harbour, you see very dynamic changes in the depth of isotherms. So in particular, these two data series are out of phase. When there's upwelling at the eastern end, there's a downwelling at the western end. Some really striking examples of that on day 205, where you see the cold blue water going all the way to the surface of the east end, while the warm water goes all the way to the bottom. So we see the same thing in the oxygen profiles. So we'll look particularly just at the west end now. And here you see a record over the most of the summer, and you see these massive swings in oxygen. As the thermocline goes up and down, the oxygen profile oscillates between having very good oxygen through the water column to almost no oxygen through the water column. And so at the same time, there's a station here that tracks the depth that walleye are at. And if we put this data on the top, you perhaps can see that the walleye are avoiding actively those regions of low oxygen. So they're primarily associated with the blue regions. Now we can quantify this in more detail by making a graph where we plot the depth of the fish versus the depth of the thermocline or the oxycline. So on the top, I've got two scenarios that could be happening. The fish could be on the left, actively avoiding the thermocline. So they'll use the depth range is limited by, at the bottom, by avoiding that oxycline. On the right, perhaps they don't care about it at all, and their depth is not affected by the depth of the oxycline. So on the bottom, this would have two quite different distributions. On the left, we would see, if they are avoiding the oxycline, all the points would be above that 45-degree line, whereas on the right, we'd expect to see a random scatter. So what do we actually see in the data? As you might expect, the walleye are avoiding the oxycline. So all the observed data that Jill Brooks found is located above that 45-degree line. They're all avoiding the depth of the thermocline. So that makes sense. Now, what happens in a cold lake where, for a cold water fish, where the fish are trying to stay below the thermocline? It would look much the same as the previous graph, except now all the fish observations would be below the thermocline on the left, whereas if they weren't paying any attention, it would look like the picture on the right. So which one does it look like here? So Kenmanville Bay also has large amplitude internal waves. It's a larger system, so the amplitudes can be quite great. Here's some data from summers over 3 different years, and you can perhaps see that there are some periods where from over a period of a couple of days, the thermocline changes depth by as much as 15 meters. So in 2015, my colleagues in the government went out and took hydroacoustic surveys during a period where there was a very large upwelling of cold water almost to the surface, and we could see what happened to the fish. So here is in detail the 4 nighttime surveys during that period. And you see the depth of the thermocline changes from about 16 meters depth up to about 14 meters depth. And on the bottom panel, there's a picture of the oxygen. There's a metalimnetic minimum in oxygen, but it's well above that 3 milligram per liter level. So it turns out not to affect the fish very much. So I would like to compare where are the fish on surveys 1, 2, 3, and 4, and are they systematically changing in response to the depth of the thermocline? So here's the first data showing the fish abundance as a function of depth. So on the bottom axis is the abundance, on the top axis is the values for temperature and dissolved oxygen. So that blue curve in particular is important. Where the blue dot is, that's the center of the thermocline. And there's a large abundance of fish. The scale is in tens of thousands of individual fish, and we can separate it up by species. Most of the fish are cisco in certain year classes. And if you look visually, you'll see on the 4 different days, generally the fish are located below that blue point. So if we plot it the same way as the previous data, Brian finds for this lake that the vast majority of the fish do indeed respond to where the position of the thermocline is. This cold water fish limits its depth to always be below the 12-degree isotherm, which corresponds pretty closely to the thermocline. And there's 4 days of data set here: pink, green, blue, and cyan, and they're all located below that thermocline. So that was interesting. We saw that cool water and warm water fish both respond to the depth of the thermocline. An interesting next step would be to expand this over the whole Great Lakes. And how we could do this is this large collaborative project called GLATOS, which has an amazing coverage of fish receivers that are all recording where the fish are and what the temperature is. My colleagues at the Ontario Ministry of Natural Resources recently installed 30 receivers in Lake Simcoe, so we can continue this in Lake Simcoe. And I'm part of a group that's installed 13 thermistor chains around Lake Ontario, so we'll be able to hopefully look at large internal waves around Lake Ontario. So today I've just been talking about fish, but internal waves are going to be present throughout all these lakes, and they're going to structure other processes apart from just fish. Plankton and zooplankton will also be strongly affected by this. So these internal waves are of great interest. So thanks for your attention. If you'd like to read more about this, these are 3 papers that have been published, and there's a 4th paper by Jill Brooks that's hopefully under review. All our collaborators on these projects from various universities and government agencies also listed here. Thank you very much.